Thank you to the conference organizers for this excellent opportunity. And I'm really excited to tell you about my work using dynamical models to both explain and predict experimental gene expression dynamics. And I think the talk before was an excellent introduction. But one of my major frustrations when working with gene expression data is that it's not often collected in a way where we can build quantitative and testable models of biological systems. So if we look at a typical microarray or RNA-seq experiment, we have a group of samples in the control condition and a group of samples in a treatment condition. And we can find genes that are upregulated and genes that are downregulated. And with these differentially expressed genes, we can then look for things like enriched gene ontology terms, or we can do something like gene set enrichment analysis to find pathways that are enriched in this data set. But we don't often get an understanding for how this system is going to behave in a new context. So if we change the type of treatment, or if we change the conditions that we measure underneath. The second issue that I have with this type of data is that we often ignore the genes that we can't identify differential expression in. And so I want to fight for these little guys over here that we're often ignoring. It makes sense in a typical setup that we wouldn't look at these genes because we can't identify differential expression between these. However, if the measurement of the treatment samples occurs along time periods after the treatment is applied, several hours, several days, then we know that we can miss transient information that's encoded in the gene expression. And we also know, even with genes that might be differentially expressed, if we don't measure intermediate time points, then we're not going to be able to identify when the gene expression changes occur, if the gene is a late responder or an early responder to the treatment. So to overcome these challenges, we developed a method where we pair gene expression dynamic data with in silico models, and I'm going to walk through how that works. So if we take the extra time up front to get high quality gene expression time course data, we can then create a library of small dynamical models that may possibly explain this behavior. And we can create simulations that match the experimental conditions we conducted. Next, we can find the subset or the family of models that match the expression dynamics that we observed in our experiments. But what's really powerful about this is we then have a set of quantitative predicted models that we can then test in other experimental contexts. So we can predict how they'll respond to a new perturbation or to a new knockout, and we can see if our predictions are going to be correct. And that will allow us to update the model. And I want to highlight that this method is very generalizable. So if you have different experimental conditions, you can change the way the data is simulated, or you can also change the type of dynamical model that you use. But to anchor my talk, I'm going to talk about how we use this method in a microarray expression data set where we looked at the regulatory role of a protein called Sprouty. For probably many of you who are unfamiliar with Sprouty proteins, they're a family of genes that have tumor-suppressing activity. They're all characterized by having cysteine-rich domains near their carboxy termini, and they're critical during development. So if they're knocked out, it can be embryonically lethal. We know a lot about what Sprouty proteins aren't, so we know they're not kinases, and we know they're not transcription factors, but we do know that they're inducible feedback regulators of receptor tyrosine kinases. And we wanted to explore the role, the transcriptional role of Sprouty in response to one of these RTKs, fibroblast growth factor. So our collaborators collected a time series measurements using microarrays of gene expression in mouse embryonic fibroblasts. We have two genetic conditions in this, Sprouty wild type and Sprouty 1, 2, 4 knockout. So we knocked out 3 of the homologs. And you can see on the right, measurements were taken at 5 time points after FGF stimulation in triplicate, going from 0 when the treatment was applied all the way up to 4 hours after the stimulation. And overall, we have around 6,000, a little over 6,500 genes that are significantly differentially expressed at least one of the time points. And I've row normalized the values here so that it's a little bit easier to see the trends, that when we sort these by when the differential expression occurs, we get a nice structure to this data. But the real question I want to ask with this data is, for any given gene, if we have the trajectory of the gene expression for the wild type, which is shown in orange, and the knockout, which is shown in green, and the shaded region is showing the confidence interval that we have from our replicates, can we find a small system of differential equations that can explain and really fit to this, this data that we saw experimentally. So to do this, we're first going to generate a library of all possible minimal systems of small differential equation systems. And these will be restricted to 3-node systems. One of the nodes is going to represent Sprouty. The other one is going to be Y, which is going to represent our output node that we can then match to the experimentally measured gene expression dynamics. And we also include a free node X, which gives us a little bit more complexity in our simulations and is also where the perturbations is going to be applied that will mimic our FGF treatment. The interactions between these nodes are signed, so they can either be activating or inhibiting. And if a node has 2 coincident edges, so for example, X, both X and S are regulating Y, then we also look at the combinations of them interacting as either additive or multiplicative edges. So overall, we have a little over 2,000 unique dynamical models in our library that we're then going to match. And we simulate data to mimic the experimental conditions that we used when gathering the gene expression data. So we have wild type, where the Sprouty node exists in our system, and we have the knockout, where its transcription rate is set to 0, and it therefore can't interact with X or Y. And because for any given gene, we don't know how FGF stimulation will affect its expression, or we look at all of a range of different possible values for the perturbation applied to node X. So it can either be activating and increasing the expression or inhibiting and decreasing the expression of X. And so when we run these simulations forward, we generate in silico data with trajectories for all X, Y, and Sprouty, and these match to the experimental conditions that we measured. So for each gene that we identified differential expression in our microarray experiment, we can then create a ranked list from our library of the small networks that best match the gene expression dynamics that we observed experimentally. And we rank these in 2 ways. The first is to match the shape of the experimental data. So for each in silico simulation, we match its mean trajectory for the wild type and the knockout, and if it is a good match, it'll be higher up in our list. And we also want to, because we have the replicates, we also want to match networks that have good differential expression and good differential expression matching to our gene expression. So for example, in the top right, this simulated data is, it's a very tight mean and there isn't as much variation, whereas in the one on the right, there's a lot more variation, so our confidence intervals overlap, and so this would be a worse match for our measured gene expression data. And to give you a flavor of how this works, I'm going to highlight and focus on 2 genes in our data set just because they were pretty interesting. They're P21 and MDM2, and they have really interesting differential expression profiles in our data set. And there's some evidence that through RAS and ultimately PI3K, Sprouty may be a regulator of both P21 and MDM2, but they're critical cell cycle regulators, and they have this really cool difference between the knockout in green, again, and the wild type expression in our data set. And interestingly, they both match best to one particular network, which I'm showing up here. And if we look at the data for the simulation that we simulated using this dynamical model, it looks something like this. And the output node Y, if you look, it qualitatively matches very well to both MDM2 and P21. But there's a lot more that we can do because we have this quantitative dynamic model that we've produced. So we can see how well it stands up to other data. So what we looked at is, and this is the same data as before, but because it's an in silico model, we can sample a lot more frequently than we can experimentally. And what I've done is, if we measure it every minute, I've downsampled the data so that it matches to our experimental conditions. And this is how the network responds to a positive perturbation on the X node, which is what I showed you before. But if we look at this, when we have, what's really cool about this, in a different experimental context, this network shows periodic behavior when no perturbation is applied, and when we actually look at all of the measurements in our simulation. And this is really cool because this is the exact behavior that we would expect of P21 and MDM2, but we did not measure this in our experiment. So what that means is we've recapitulated the behavior to both match what we see experimentally and also to predict and behave the way we would expect in other contexts. So to summarize, I hope that I've shown you that if we have really nice comparative gene expression time course data, we can generate a library of possible matches to this, and we can then evaluate all of these to see how well they match. And if we do a good job matching these, we can find small dynamical models that explain the data we saw experimentally and also generalize into other contexts. And this is really powerful because now we have a model that's both quantitative and testable. So if we want to see how it behaves in a different context, we can go and generate experimental data to validate this and also update the model if we find that there are some differences. All of this work would not be possible without the incredible help from my lab, the Models and Bagiri lab. I do want to highlight that my lab mate, Jia Wu, does have a poster in the second room. She's very happy, and she'll answer all of your questions. My advisor, Netta Bagiri, has been incredibly helpful in providing wonderful guidance for this project. Our excellent collaborators are Dr. Banam Nabet, who's now at Dana-Farber, postdoc at Dana-Farber, and he collected all of the microarray data, and his former advisor, Professor John Licht, who's been incredibly supportive in allowing me to explore this data set. And of course, thank you to all of my funding sources. I want to highlight again that there's a lot we can learn from dynamic information. I didn't get to show you all of it today, and with that, I'm happy to take your insightful questions, or I don't want to get in the way of you guys in coffee, so speak to me afterwards if you want as well. Thank you.